Hello friends, I am Shinde. In this video, we are going to discuss about the monocot leaf, means the anatomy of monocot leaves. In last lecture, we have discussed about the dicot leaves. The dicot and monocot, this difference between these two leaves is main difference of these two leaves. The dicot leaf, it is both the surfaces of the leaves, they are different or dissimilar. They can easily identify in dicot leaves. And this leaf, it is also called the dorsiventral leaf in dicot plants. But in monocot plants, it is a difference between these leaves. This monocot leaf, it is isobilateral leaf. In monocot plants, it is a isobilateral leaf. This iso means similar. In this monocot leaf, it is both the surfaces of similars. They do not identify. In monocot leaf, both the surfaces of the leaf, they are similar structure. But in dicot, these leaves are dorsiventral, because dorsal and ventral surface are easily identified. But in this monocot leaf, these both the surfaces are similar, they do not identify. Because in this leaf, the main midrib, this middle vein is present, and this middle vein, the veins and veinlets, they are parallelly run. Means this parallel venation of the leaves in monocots, and it is a similar veins. But dicots, the upper surface or dorsal surface is a darker. But in monocots, these two surfaces are green color. They do not identify. Now this anatomy of this monocot leaf showing the different parts in this internal structure of monocot leaf shows the different layers, and this monocot leaves, there are upper epidermis and lower epidermis. This epidermis, it is a single layered structure, and this epidermis, the upper epidermis, it is called adexile epidermis. This upper epidermis, it is called the adexile epidermis. Adexile means it is the stoma is not found in this epidermis, and this epidermis, it is a adexile epidermis, means stomata absent. But in the lower epidermis, this lower side, this stomata get present. This stoma are present, means stomata is present in the lower surface. This epidermis, abexile epidermis. And this epidermis layer, these are covering of this epidermis, it is found the thin layer structure, it is called the cuticle. This cuticle is a protective function of the cells, and this cuticle is also found in the upper or adexile epidermis or the abexile epidermis. But in adexile epidermis, the variable cells are present. These are the variable cells, and these cells are called bulliform cells. These cells, they are variable in the epidermis, and these cells are swells. This swelling of these cells, because these cells enter the water, and these cells, they get swell, and these variable cells, bulliform cells, are most important cells are found in the leaves, because most important function of these cells, it is this fill in water in these cells, and most important for rolling of leaves. These cells means leaf is rolling with the help of these cells. Bulliform cells is important because these cells, they can fill the water, and these cells are important for rolling of the leaf as well as stretching the leaves in monocot plants. It is the most important function of these epidermis cells or bulliform cells are found in this region. Now in dicot and monocot, the internal structure is same. Means dicot leaf, they are present the mesophyll cells, but these mesophyll in dicot, they are divided palisades and spongy tissue. But in monocot leaf, only the mesophyll cells are present, inner surface, inner side, and they do not have the palisade and spongy tissues. Only the similar cells, all the cells are mesophylls, parenchyma cells. These mesophyll cells are found in the inner side of the leaf, and this leaf is totally filled with these mesophyll cells. But innermost, they are rounded structure, it is called the vascular bundles, and this vascular bundle is found in the mid-rib region of monocot leaves. This vascular bundle, these cells, they are bundle sheath cells. These cells, with these vascular xylem and phloem cells, they are surrounded by the bundle sheath cells. These bundle sheath cells, and these bundle sheath cells, their innermost cells are found, or inner part, it is a protoxylem cells and metaxylem cells, or also called the metaxylem vessels and protoxylem vessel. But these cells also located, these are present the inner surface of the bundle sheath cell. The vascular bundle of monocot leaf is a distinct as a dicot leaves. In monocot leaves, this vascular bundles is conjoined, collateral and close type, because in this region of the leaf the cambium is absent, means secondary growth is absent in the monocot, and in the internal structure of bundle sheath cells, with the vascular bundles, this cambium is totally absent, and this type of the vascular bundle, it is conjoined, collateral and close type of the vascular bundles. In monocot leaves, the mesophyll cells, they do not divided into palisade and spongy, and these vascular bundles are present, the conjoined, collateral and close type of the vascular bundle. The next one, this lower epidermis is the abaxile epidermis, they showing the small pores, and this pore, it is called the stoma. In the leaf, number of stomata are present, but these pores are, number of stomata is present in the lower surface of the leaves. As compared to the upper surface, the number of stomata is present in the lower surface of the leaves, and this stomata is most important function for gaseous exchange as well as this transpiration. These two important function of the stomata. In these small pores is found in the abaxile epidermis, and this pore is helpful for the exchange of gases, and this exchange of gases is takes place in this substomatal cavity. This is a ear space is found in this region. With this ear cavity is present, and this substomatal cavity, it is present the lower surface as well as the upper surface. This stomatal cavity is found in the monocot leaves. Now this is a structure of face of monocot leaves. In the anatomy of monocot leaves, in the internal structure of this leaf showing two important parts, epidermis, this abaxile and adaxile epidermis. This outermost layer is found in the leaf, and this layer, the most important cells are present. These cells are larger in size, thin, old cells, and these cells, it is called the bulliform cells, and most important for these cells for in monocot plants for stretching, as well as it is important role for the rolling of the leaves. These cells are most important for the rolling of the leaves, and the internal structure is similar as a dicot leaves, but these mesophyll cells, they do not divided into palisade and spongy tissues, only the mesophylls. But in most important for these mesophyll cells, they are present the chloroplast cells. Large number of chloroplasts containing cells are present in each cell of mesophytes, and these cells, they have present the chlorenchymatous cells. These are the chlorenchymatous cells are present in the mesophytes because the presence of chloroplast. This number of chloroplast containing cells are present in the mesophytes cells, and this chloroplast is important for photosynthesis. In the mesophytes, number of chloroplast containing cells are present, and these cells are called chlorenchyma. And this, for example, in maize, in monocot leaves, for example, maize, and this maize showing the C4 plants. Means it is a C4 plants. The anatomy or anatomical peculiarities of these maize plants, internal structure of these maize plants, it is to explain this is a C4 plants and showing the crans anatomy. In the monocot leaves, they are present the bundle sheath cells and mesophyll cells. The bundle sheath cells, they are absent a granule chloroplast containing cells, but in other cells, mesophyll cells, they are present the granule chloroplast. In this leaf, the C4 plants means maize, they can explain the crans anatomy, showing the difference between these two cells. Means one cell present the chloroplast containing cells, grana is present in the mesophytes, and bundle sheath cells, this grana is absent, and these cells are agranule, and it is the most important dimorphism is takes place in these maize plants. This is the internal structure of monocot leaves. We have completed this monocot leaf or internal structure of monocot leaf.